నమస్కారం. డాక్టర్ బిఆర్ అంబేద్కర్ సార్వత్రిక విశ్వవిద్యాలయం విద్యార్థులకు స్వాగతం. ఈ ప్రసారంలో
After knowing the advantages and the disadvantages of various culture techniques, the most probable methods throughout the India as well as in other countries, most probably following outdoor and the closed systems and the open pond system, even though some disadvantages are available. And these systems are helping in getting a good yield to the farmers as well as to the consumers. And inoculation method is also very important for getting a good yield from the culturing media. Harvesting should be prompt and we should follow all the protocols what I said in the above lesson. And after realizing in the first lesson the importance of algal cultivation as well as the cultivation methodology and what inoculation vessels we have to utilize in the laboratory and in what way the harvesting methodology we have to utilize for getting a more yield in case of algal cultivation. In the second lesson, again, the benefits of cultivation of algae realization is very important to the peoples. And why to do this cultivation of algae? What are the uses of algae? And in future tense, what utilization may takes place for the common peoples and to the farmers and to the consumers, and in what way it is pruning the jobs in future, especially whoever are pursuing their MSc in the botany. Especially algal cultivation is also, phycology is a special area where the cultivation of algae is giving several chances to the peoples, educated peoples as well as uneducated peoples, even in the industry level as well as the pharmaceutical level, and also at the level of agriculture we are using this algae. That's what algal cultivation is very important to the society. Let's start the benefits of cultivation of algae in this lesson. Later on we can see some of the important algae which had been utilized for the SCP point of view, that is single cell protein point of view, and as a biofertilizer, as well as in other countries, not in our country, in other countries, one of the macroalga porphyra habit had been cultivated. Spirulina cultivation. This is one of the organism which is especially having a SCP constituent is more when we are comparing with the fishes and the beef, especially protein qualities which are very abundant in the animals. Those proteins are higher range in case of spirulina. So therefore I want to put forward regarding the spirulina cultivation in detail, that is algal tanks, especially we have to prepare circular or rectangular cemented tanks are constructed, and the size may be according to the yield needed. How much of the yield is needed, that much of the measurement is needed for the making of the circular or the rectangular cemented tanks. And depth should be about 25 centimeters. Open ponds are suitable for especially tropic and the subtropical regions. Light intensity should be low because it may be leading the photolysis of the organism. Spirulina, especially exposed to the high light intensity, may lysis the cells also. It is a very difficult condition if we are maintaining the high light intensity. And temperature should be range in between 35 degrees Celsius to 40 degrees Celsius, optimum temperature it is, and the maximum growth will be takes place at this particular temperature. Next, coming to the pH. Spirulina grows mostly highest pH ranging from the 8.5 to 10.5. Of course, 8.5 should be maintained at the initiating of the culturing, whereas automatically we have to elevate it to the 10.5 pH maximum. And spirulina cultivation next followed by the agitation. Agitation of the culture is very necessary to get a quality and the better density and the yield from the culturing medium. The culture is agitated by the brushes or the paddle power, pipe pump, wind power, rotators, etc. Harvesting, the filaments of the spirulina float on the surface of the water forming it is forming a very thick mat on the surface of the water, and it can be harvested by fine mesh screens or by the nylon or the cotton cloths, etc., which is a very cheapest method also. And a drying method followed by it is the thin wall. Due to this drying method, why we are following the sense, spirulina cell wall is very thinner comparing with the other alga. Sun drying is a very cheapest method and the most suitable, and it is recommendable and economical also. And yield, especially from this method, an average yield of 20 tons per hectare per annum has been obtained in India and other countries also. Next, spirulina is one of the ultimate food. Cultured for over 600 years, and it having a richest protein, as I said previously, 65 to 68% of the protein, almost all similar to herring fish. 1 acre of this stuff produces 10 tons of the protein, almost all what we are not getting from the wheat, that is only 0.16 tons only from the wheat. What we are gaining from the spirulina is more productivity and the more protein content, which is very useful for growth and the development of the living organisms on the earth. And other goodies are also available. My meaning is goodies is nothing but other algal member, not only spirulina. And this chlorella and Senidesmus are also excellent sources of the single cell proteins. Could yield 40 tons per acre per year. 3 feet deep is essential for the cultivation of chlorella and Senidesmus, and 1,000 cows can feed on this particular algal members for a year, just for an year. That's it. And use of Spirulina SCP. Spirulina is a single cell and spiral shaped blue-green microalgae. Highly digestible food it is. 60% of the vegetable protein which is pre-digested by the algae only. It is higher in protein than any other foodstuffs. 1 teaspoon of spirulina contains 280% of beta carotene and 110% of B12 vitamin, 15% of iron and 2% of calcium, and no fat is present in spirulina. That is a very good utilization of spirulina, especially in our country and other country. It's a outstanding nutritional profile if we are looking after. It is possessing fatty acids, especially lipids, the nucleic acids, RNA and DNA, B complex vitamin, vitamin C and E is also available. And carotenoids, chlorophyll, especially if we are consuming this as a food and feed, it is one of the blood purifier. And also phycocyanin is one of the blue pigment existing in this blue-green alga, which is a protein that is known to inhibit the cancer. So, it is one of the wonderful utilization of the spirulina. Phycocyanin is one of the wonderful pigment which is not at all present in any other algal member. And next, coming towards the carotenoids and chlorophyll may also contribute to the spirulina as an anti-cancer, and also apparent immunogenic effects are also it is showing. Spirulina is having a 2 to 6 times richer in B12 vitamin than nearest raw beef liver also is nothing in front of the spirulina. And spirulina is 58 times richer than raw spinach in case of iron constituent. Spinach is also not giving that much of the iron, my meaning is. And if we are consuming the spirulina, we can get a 58 times better iron proportion from the SCP value of spirulina. Spirulina is nature's richest whole food source of vitamins, as well as it is 3 times richer than the raw wheat grains, as well as biologically active, and it is having a 49% of greater vitamin value. Spirulina is many times richer than alfalfa or the wheatgrass. Spirulina is nature's richest whole food source of complete high biological value protein. Spirulina possesses 60 to 70% of protein constituent when we are comparing with other foodstuffs such as soybean having only 35% and beef having 18 to 22% of protein, and eggs having only less constituent 12 to 16%, and tofu only 88%, milk having 3% of the protein. Then with the help of this particular information, spirulina having a higher proportion of SCP when we are comparing with our regular eating items such as egg or the tofu or the milk or the soybean or beef. And mass cultivation of cyanobacterial biofertilizer. This information I want to give especially for the farmers and who having especially difficulties in handling with chemical fertilizers. For outdoor mass cultivation of cyanobacterial biofertilizer, the regional specific strains should be utilized. Mixture of 5 or 6 regional acclimatized strains of cyanobacteria, that is species of Anabina, number 1, Alocida and Cylindrospermum, Gliotrichia, Nostoc, Plectonema, Tolipothrix are generally used for starter inoculum. These are the 6 strains should be what inoculated at a time in the culturing media. 4 methods are utilized. One method is cemented tank method, second method is shallow metal troughs method, and polythene line pit method, field method. Among this, pit line method is the most probably utilized throughout the world. The polythene line pit method is most suitable for small marginal farmers to prepare algal biofertilizer. The protocol of this biofertilizer production is prepare the cemented tanks, shallow trays of iron sheets or polythene lined pits in an open area with less than 1.5 meters and transfer 2 to 3 kgs of the soil and add 100 grams of superphosphate to it. Water the pit to about 10 centimeters height. Mix lime to adjust pH 7 and add 2 ml of insecticide, that is malathion. Why to use malathion? This malathion will kills the mosquito larvae in the open pond system or the closed ponds. Mix well and allow to settle down soil particles. When water becomes clear, sprinkle the starter inoculum. My meaning is 6 strains what I had said above should be on the surface of the water. And optimum growth of cyanobacteria is achieved at the temperature between 35 to 40 degrees Celsius. Maintain the water level about 10 centimeters until and unless this entire procedure may not be finished. After drying, the algal mat will get separated from the soil and forms flakes, and these flakes are collected, powdered and kept in a sealed polythene bags and supplied to the farmers. And nostoc spaces, especially this figure is showing a nostoc, which is also a very good biological fertilizer, and BGA biofertilizers nowadays available in the markets in a commercial way. And next, cultivation of porphyra, which is a macroscopic alga. Actually, it is a marine, it is existing in the marine habitat, having a thallus-like plant body, no root, no stem, no leaf, as I said at the beginning of algal characteristic features. Cultivation of this porphyra is a very essential, and in these figures especially I am going to visualize you how the maintenance of culturing method of porphyra will be done on the earth also by utilizing the sea water in closed systems, and harvesting method and utilization in the laboratory in the third figure, how they are preparing the flakes of porphyra and distributing to the various commercial and the industrial levels. Cultivation of the porphyra had been begun in the 17th century in Japan, Korea and China, and since become one of it is most important industries are also established on the basis of the porphyra using shallow water areas in all of these countries. Main producer countries are China. This map is showing you the main producers as per the fishery statistics of FAO is indicating China is a major, and Japan is also involving and participating in the production of the porphyra species. Cultivation of the porphyra following some of the methods. Number one is floating system. Floating system is this method is used in Japan and has recently it had been widely adapted by the Chinese peoples, and the nets are especially attached to the bios floating on the surface of the sea water, so that nori is a common name where these Japanese peoples are using this porphyra to be pronounced. Actually, porphyra may not be understood by the Chinese farmers and the Japanese farmers, so therefore it is pronounced as a nori. Nori thalai is constantly soaked to the water due to this floating system. With this method, it is possible to cultivate the thalai even in areas outside the shallow bays. Next, cultivation of the porphyra is also following semi-floating system and also fixed net system. And the semi-floating system, in this method, a mixture of the floating and the fixed net systems we are utilizing. High tide, when the high tides are taking place, we are using this method, and when the low tides are taking place, they may be float on the surface of the water, and this system is especially applicable where we are cultivating this porphyra on the land. This contains the advantage of fixed nets and floating systems, so it has been widely adapted in China. And fixed nets, in this method, especially also known as pole system, in this nori had been poled by using some of the 4 poles towards the surrounding this system, and nets are exposed to air, the nets become dry, and intertidal pole cultivation is also often preferred over the floating and semi-floating systems. Floating cultivation in deep water, because it predicts the exposure of the proper duration, which helps to reduce the incident of diseases and other predators grown competing with the weed species, especially epiphytic diatoms. However, this type of cultivation is restricted to the inner portion of bays with shallow and sandy bottoms. And freezing nets are the last method of cultivation of the porphyra. This method has 2 advantages: preventing diseased nori and improving the quality of the final product. And procedure for processing the freezing nets is when the young thalai is reached to the 1 to 3 centimeters long, they are brought back from the culture area together with the nets are dried in the air until the moisture content of the thalai decreases by 20 to 40%, which usually requires 2 to 3 hours. Dried nets are put into vinyl bags, and dried nets are stored in a freezed condition at minus 20 degrees Celsius. When required, the nets are brought back to the field for rearing purpose. Harvesting techniques: after rearing in the open seas for 40 or 50 days, in the especially when cultivating the porphyra haitensis and porphyra yazonensis are the 2 species respectively. First harvesting phase begins and harvesting achieves to the 4 months of the time. After 5 months of the time, when we are achieving, the intervals of the harvesting is every 10 to 15 days like that. Therefore, the crop is harvested almost 10 to 12 times annually. And two methods are used, namely manual and the mechanical method in harvesting, handling and processing method. The harvested thalli are cut into pieces and processed by the traditional forms. A thin rectangular flakes are prepared. Each will be having a minimum size and weighing about 3 grams. The procedure consisting of selecting the wet nori, washed with the seawater and later on washing with the fresh water, cutting it and softening and making the flakes, dehydrating, drying, grading and packing the dried nori can be refined into seasoned products that can be directly consumed. These items have become popular as sweets called as a candy in other countries, Japan. Production cost is unexpected, no information is available for this supply, and dried nori can be refined into seasoned products, then directly it can be consumed. These items have become popular as a sweet candy. And this particular picture is showing you how to harvest this particular porphyra from the seawater, and as I said, the methods of harvesting above, and the porphyra sweets are called as candy in the Japan country, and as well as in China, wherever they are cultivating, they are called as a candy, not called as a sweets, but they are almost all reliable to the sweets. Algal culture collections. Specific algal strains acquired from algal collections. Over 500 culture collections registered with the World Federation for Culture Collections. Products of algae culture. Especially algae will be utilized in various areas. Number 1, industry. Agar-agar produced from the red alga, example Gelidium gracilaria. And alginates are obtained from the red alga from sargassum, and as well as macrocystis. Creams, jellies and ice creams, soups, sauces are also prepared from the alginate. Iodine produced from the brown alga Phaeophyceae. Example laminaria is a best example, and it is a very good treatment for the goiter, simple goiter. Bromine. Bromine is obtained from the Polysiphonia, which is utilized commercially. Carrageen is obtained from the Chondrus crispus. Shaving creams are prepared, and shampoos and other shoe polishes are also prepared from the carrageen. And next, in case of agriculture, nitrogen fixing blue-green alga, Anabina, Alocera, nostoc, Cytonema, Tolipothrix, etc. Can enrich the content of nitrogen in the soil as a biofertilizer. And as a food also we are utilizing nowadays throughout the world. Chlorella, which is 50% of the protein, 20% of the lipid, carbohydrates and vitamin A, B, C are also available. And next, porphyra is called as amanori in Japan. Common name is amanori in Japan, and it possesses 35% of the protein, 45% of carbohydrate, vitamin A, B, C, E are also available in porphyra. And next, laminaria, alva and calarpa are useful for the food preparation point of view. And next, fodder point of view also we are using as some of the seaweeds such as Phaeophyceae and Rhodophyceae members for the animals especially. In the case of medicine field, we are using this algae, chlorella, used as an antibiotic, and next, cladophora utilized as an antiviral, and laminaria useful for especially blood anticoagulant, and also agar-agar useful in the preparation of the pills and the ointments in the pharmaceutical industries. Oils and gases are also produced from the sargassum and laminaria, Batriococcus, Spirulina, and these all of the algal members are producing biogas to reduce the environmental pollution nowadays.
ఈ కార్యక్రమాలపై మీ అభిప్రాయాల్ని, సూచనల్ని, సలహాల్ని ఆహ్వానిస్తున్నాం. మా చిరునామా ద డైరెక్టర్, ఆడియో విజువల్ ప్రొడక్షన్ అండ్ రీసెర్చ్ సెంటర్, డాక్టర్ బిఆర్ అంబేద్కర్ ఓపెన్ యూనివర్సిటీ, ప్రొఫెసర్ జి రామరెడ్డి మార్క్, రోడ్ నంబర్ 46, Jubilee Hills, Hyderabad 500033.